Good day everyone. This is Miss Seleni N. Onelongo and I will discuss about the genetics of cancer.
Cancer is a genetic disease at the somatic cell level. Cancers are a large family of diseases that involve abnormal cell growth with the potential to invade or spread to other parts of the body. The image shows a human melanoma cell undergoing cell division. The chromosomes, which is the color blue, have separated and the two daughter cells have almost split apart. Only a small breach of cytoplasm remains. The green staining labels the endoplasmic reticulum and the red labels the mitochondria. All tumor cells shows the six hallmarks of cancer. These characteristics are required to produce a malignant tumor. They include cell growth and division absent the proper signals, continuous growth and division even given contrary signals, avoidance of programmed cell death, limitless number of cell divisions, promoting blood vessels construction, invasion of tissue and formation of metastasis, and the progression from normal cells to cells that can form a detectable mass to outright cancer involves multiple steps known as malignant progression. Cancer is caused by mutation, the basic mechanism in all cancer. Mutations or damage to the genome can be caused by mistakes during normal DNA replication. The DNA can be attacked by free radicals, ionizing radiation, UV radiation, and chemical carcinogens, producing mutations in somatic cells. Mutations in cancer genes can occur somatically or can be inherited. Mutations in some cancer genes can be inherited from parents or known as germline mutation, in which case they are present in every cell of the body. Such people are at a higher risk of developing cancer. Somatic mutations can occur in any of the cells of the body except the germ cells or the sperm and egg cells, and therefore are not passed on to children. These alterations can, but do not always, cause cancer. There are two types of cancer genes, the tumor suppressor genes and the oncogenes. Tumor suppressor genes are the TSG code for proteins that slow down cell growth. They can halt the cell growth cycle to stop unnecessary division or promote apoptosis or the cell death if the cell's DNA is damaged. There are different tumor suppressors proteins carry out for the following functions. First, repressions of genes which are essential to the cell cycle, therefore inhibiting cell division. Second, linking the cell cycle to DNA damage. If there is damage to the cell, it will not allow it to divide. And the third, identify where the DNA damage is irreparable and initiating apoptosis or what we know as cell suicide. The animated chromosome diagram illustrates that both copies of the TSG have to be inactivated by mutation or other alteration for there to be a loss of cell cycle control. If one function copy remains, there is still a break on the cell's growth. The cell is a car energy. One way to think about tumor suppressor gene is to see them as the brakes of a cancer. There is a genes on both chromosomes, so in a sense there are two brakes. If one gene is mutated and its protein loses its functions, the cell can still halt and prevent unregulated cell growth, as the other copy of the gene, or brake, is still functioning. However, if that backup copy also changes and no longer codes for a functioning protein, the cell cycle is no longer under control and this can lead to cancer. Proto-oncogenes code for proteins that drive cell division. When these genes acquire mutation that result in continually active proteins, they become oncogenes and cause uncontrolled cell growth and division. If you are using the car analogy, oncogenes can be seen as the accelerator. When one healthy copy of the proto-oncogenes is altered, it is the equivalent of the accelerator pedal being stuck, speeding up cell growth and division. Notice in the case of oncogenes that it only takes one copy of the gene to undergo changes to lead to cancer, rather than both copies as is the case with TSGs. There are four classes of normal regulatory genes are the principal target of genetic damage includes first, the growth-promoting proto-oncogenes, second, the growth-inhibiting tumor suppressor genes, Third, genes that require programmed cell death or the apoptosis. And the fourth one is DNA repair genes. Proto-oncogenes have multiple roles participating in cellular functions related to growth and proliferation. Mutations convert proto-oncogenes into constitutively active cellular oncogenes that are involved in tumor development. There are two types of proto-oncogenes. First is the cellular oncogenes or the C oncogenes. It is a proto-oncogenes which have been to mutate in any individual. Unlike normal oncogenes or the N oncogene, which is a proto-oncogenes that have not been found to mutate. Oncogene is a gain of function mutation because the cells with the mutant form of the protein have gained a new function, not present in cells with the normal gene. Oncogene is a dominant or activation of one allele is sufficient. When a proto-oncogene becomes activated, it is called an oncogene. When an oncogene becomes activated, it might cause cancer. When genes implicated in the control of cell cycle repair DNA mistakes and tell cells when to die. The product of tumor suppressor genes normally lack abnormal growth and malignant transformation and lead to malignancy when the function of both alleles is lost. When tumor suppressor genes don't work properly, cells can grow out of control, which can lead to cancer. In contrast to mutations in proto-oncogenes, which are dominant in their action, most mutations in tumor suppressor genes are recessive. DNA repair genes are genes that fix any mistakes made when the DNA is replicated or copied. Mistakes that aren't fixed becomes mutation, which may eventually lead to cancer. For example, genes responsible for HNPCC or the hereditary non-polyposis colon cancer. When these genes do not repair the errors in DNA, HNPCC can result to what we know as cell suicide genes. Cancer development is a gradual process where genetic changes are accumulated over a period of time. The different stages of cancer progression are shown. First, we have the benign tumor. A cell begins to divide more rapidly than its neighbors and a localized mass of cells forms. Next is in situ cancer, wherein a cell begins to divide in an uncontrolled way. Then we have invasive cancer. The tumor gets bigger and cell begins to invade surrounding tissue. In this case, the illustration is showing the tumor arising from a cell within a valley of the large intestine, expanding outwards into the surrounding area. And lastly, we have the metastatic cancer. Tumor cells travel to other parts of the body throughout the bloodstream and deposit in other organs, leading to secondary tumors, known as the metastasis. This time, let us proceed to the external causes of cancer. First, we have the ultraviolet radiation. Natural radiation, such as the ultraviolet rays from the sun, is known to be the primary cause of skin cancer. Through prolonged, unprotected exposure to sunlight and ultraviolet light in the form of sunbeds, we increase our risk of developing skin cancer. Another external cause of cancer is tobacco smoke. The chemicals in cigarette smoke have been casually linked to the development of lung cancer. Lung cancer causes around 1 million deaths worldwide every year, almost all of which are associated with smoking. Tobacco smoke contains more than 60 mutagens that bind and chemically modify DNA. These brand the lung cancer genome with characteristic mutational patterns. Overeating leads to obesity. Obesity is known to be associated with bowel and stomach cancers. If obesity can be reduced, then the number of cancer cases will be reduced. Many cancers have a course that is highly variable and therefore difficult to predict, because cancer genes dictate the aberrant phenotype of cancer cells. Genotypic analysis can provide information on the capacity of a given tumor to grow and spread. This information can potentially be used to categorize tumors and to predict their cause and responses to therapy. First is early detection. It is the most treatable cancers are those that are diagnosed at an early stage. Then we can have a genetic counseling. It is a communication process to inform the consultants about the recurrence risk of predisposing familial cancer, which might affect the offspring of future generations. There are three important steps in genetic counseling. These are the establishment of diagnosis, estimation of recurrence risk, and communication of relevant information to the consultants in a sympathetic manner so that the latter can exercise their own choices or options available for dealing with the risk. As part of the process of genetic education and counseling, genetic testing may be considered when the following factors are present: an individual personal history, or if the family history is suspicious for a genetic predisposition to cancer. Genetic testing is done on a small sample of body fluid or tissue, usually blood but sometimes saliva. You can also have the gene therapy. Envisages the replacement of the deficient gene in the form of a protein or an enzyme or correction of abnormal genes. Another prognosis is the gene transfer. It is a new treatment modality that introduces new genes into a cancerous cell or the surrounding tissue to cause cell death or slow the growth of the cancer. No genetic disorders have been conclusively cured by a gene therapy, but some promising results are obtained from ongoing clinical trials. So before I end my report, I want to share with you the important information about cancer. Cancer is a genetic disorder in which the normal control of cell growth is lost. The basic mechanism in all cancer is mutation, either in the germline or much more frequently in somatic cells. Cancer is multifactorial diseases. Much remains to be learned about the genetic processes of carcinogenesis and about the environmental factors that alter DNA and thus lead to malignancy. I hope you have learned something about the genetics of cancer. Thank you and God bless.